Hello, my name's Greg Jeffries. I'm a program leader here at the MRC Laboratory of Molecular Biology in Cambridge. Our group in the Neurobiology division is interested in the circuit and genetic basis of behavior, with a particular focus on innate and sexually dimorphic behaviors. Our work uses most of the experimental tools available in our favorite model organism, Drosophila. This includes in vivo electrophysiology, quantitative analysis of behavior, and also genetically targeted manipulations of neuronal activity. However, anatomical studies of circuit organization and synaptic connectivity are critical to our work because they provide the bridge between studies of circuit function and behavior on the one hand and studies of genetics and development on the other. Many of our studies begin by dissecting the brain out of a fly, staining it to reveal individual neurons, and then moving to a confocal microscope in order to take an image of the brain. Over the last few years, several fly labs have used these approaches to generate very large data sets, including both images of single neurons and of genetic driver lines. These resources have been generously shared with the community and are now accelerating circuit research. There are now over 40,000 such images publicly available, and data volumes are increasing markedly with efforts to obtain complete connectomes for the larval and adult fly brain. All these data need new tools in order to query and organize them. Taking inspiration from tools such as BLAST that allow protein sequences to be compared, we've developed and validated a new algorithm that we called NBLAST that allows neurons to be compared. The first author of our study, Costa, will now tell you more about NBLAST, how it works, and what you can do with it.
Hi, my name is Costa, and I work with Greg to develop and validate the NBLAST algorithm. First, I'm going to explain how the algorithm works, and then I'll go through a couple of examples that we show in our paper. NBLAST works by comparing pairs of neurons based on their position in the brain and their morphology. In order to validate the algorithm, we use the fly circuit dataset, a dataset of over 16,000 images of single neurons that has been produced by the lab of Anqin Chang in Taiwan. And in order to use the position of the neurons in the brain, we first need to register each image to a template brain. Then each neuron is decomposed into segments, and each comparison takes one segment from one neuron, which we call the query, and compares it against the nearest neighbor in the other neuron, which we call the target. This comparison is calculated as a score that takes into account the distance between the segments and their direction. And this is weighted by a probability function that is based on if the segments are coming from the same neuron type versus if they were coming from different neuron types. So the score will be higher for pairs of segments that are closer and point in the same direction. NBLAST searches can be done in a variety of ways, and I'll go through a couple of examples. So the first thing you might want to do is to find the most similar neurons to a specific one. And in this case, we start with a whole neuron, and we query against the whole data set. And then we can order the results by score and very easily find the most similar one. Another type of search we can do is to find what neurons are present in a genetic driver line. And this type of search is very useful for researchers when they're planning their experiments because it makes it very easy and quick to find what transgenes they need to pick to label specific neuron types. And in this type of search, we're using a set of 3,500 GMR Gal4 lines produced by the Rubin lab in Janelia. Here we're searching for driver lines that label the MAL neurons. And in this case, we have a set of 10 neurons that we're using as a query, and we can see the top hit is a driver line that specifically labels this neuron type. Once we developed the algorithm, we needed a way to see if it works. And to do that, we use known cell types and using NBLAST scores, see if we could recapitulate the classification that is already known. I'll now go through a couple of examples. The first one are the Kenyan cells, the intrinsic mushroom body neurons in the fly brain. They're involved in memory and learning. And there are three major types, so the gamma, alpha beta, and alpha prime, beta prime. Our classification of the alpha beta neurons reflected the known hierarchy that's already known, and that shows that NBLAST works. For the gamma neuron type, we were able to find new subtypes that have not yet been described. Another cell type we analyzed was the P1 neurons. So these are sexually dimorphic neurons involved in courtship. what our analysis of male P1 neurons showed was that there were actually 9 subtypes we were able to distinguish, and some of these are new and not yet been described. NBLAST scores can be used to organize large data sets of neurons, and this is becoming more and more necessary as the volume of image data increases. In this case, we divided the whole of the fly circuit data set using a clustering method into 1,052 groups of similar neurons. What we found was that many of these clusters corresponded to neuron types, and this indicates that this type of approach might be useful to define what a neuron type is and as a starting point for further work. If you'd like to know more about NBLAST examples, code, or data, just go onto our NBLAST site.
For the future, I'd like to highlight 2 things. First, we've already tested NBLAST with data from vertebrates, specifically zebrafish, and it seems to work well there. Second, we've also tested NBLAST with neurons traced out of electron microscopy volumes. It also works very well with these data, and we are hopeful that NBLAST can therefore help to organize the huge amounts of data that will come out of connectomic efforts in the near future.